Hello everyone, today we are going to talk about immune system and central lymphoid organs. And before doing that, at the beginning of the lecture, we are going to cover the basics of our immunity, how immunocompetent cells do interact one with another. For example, how B cells get activated, how antigen-presenting cells do activate T cells, and so on. Because lymphoid organs are represented predominantly by lymphocytes, and to understand how they are distributed inside the organ, we have to understand how they interact one with another. What is the sequence of events in our immune reactions? So let's start. And first of all, we have to say that our immune system is represented not only by lymphoid organs like thymus, red bone marrow, lymph nodes, spleen, but also by diffuse lymphatic tissues present in mucous envelopes of different organs, predominantly digestive tract, respiratory tract, and so on. Also, immune system, it includes leukocytes distributed in almost every system of organs. So inside our connective tissue, for example, inside our brain, there is a set of leukocytes providing the immune surveillance. So immune system is ubiquitous. It forms distant organs, but at the same time it is present in every organ in form of cells infiltrating loose connective tissue and other tissues. And then also immune system, it includes lymphatic vessels collecting lymph from different organs and bringing this lymph to lymph nodes for the filtration. We are going to discuss this in our next lecture. And what is the function of immune system? It provides us surveillance and it helps to reject foreign antigens of our body. So the basic problem of immune system is how to distinguish self-antigens from foreign antigens, because self-antigens should be tolerated. We just elaborate tolerance to them. We do not fight against self-antigens, but at the same time we fight against foreigners. And how to distinguish one from another, that's the crucial problem, and that's what we are going to talk right now. So from school you have to remember that immune response should be considered as innate and adaptive. You remember innate immunity is non-specific and it is inherited, while adaptive immunity is elaborated after the first contact of antigen. So adaptive immunity, it is called so because we adopt it, we acquire it along our life. It is not inherited and it is specific. So we reject exact type of bacteria, not bacteria as a whole, but exact types of bacteria cells. And today we are to talk about adaptive immunity, especially because innate immunity is realized by granulocytes, and that's what we have discussed in the topic blood. Then immune response also could be divided into cellular or cytotoxic, when killer cells, for example, they kill another cell of our organism, and also humoral. So you have to understand what is humoral immunity. Humoral means that it is mediated by antibodies produced by B cells. So B cells themselves, they do not attack foreigners, but they produce special proteins, antibodies, which are highly specific to exact antigens. And antibodies interacting with the antigens neutralize them and help neutrophils to absorb, to phagocytose them. So in this way, antibodies, they help to eliminate antigens out of our organism. Let's move on. And now we have to talk how our immune system do analyze antigens and how it distinguish our own antigens from foreign. And in this way we have to say that major histocompatibility complex performs the main function. There are two types of those receptors. Type 1, present on the surface of all nucleated cells and presents peptide fragments of self-proteins to T killers. So what is the MHC type 1, major histocompatibility complex? This is a set of receptors destined to carry examples of all proteins produced inside the cell on its surface. So for example, if cell produces insulin or any other hormone, this insulin is broken down inside the cell into small fragments and fragments of this insulin is presented by major histocompatibility complex receptors on the surface of this cell. For what purpose? Because there are killer cells always monitoring the surface of another cells in our body. And killer cells, if they find here normal protein, characteristic for this tissue and for our organism, then it doesn't do anything. But in case if insulin is defective, if as a result of mutation we start to produce abnormal proteins, or in case of viral infection, cell starts to produce viral peptides, which are not characteristic for our organism. In this way, those fragments will be presented by major histocompatibility type 1 receptors to killer cells, and passing by and scanning the surface, natural killers or CD8 killers might recognize abnormal protein, and they conclude that this cell produces something not normal. This cell is gone crazy, so it should be killed. And in this way, killers, they stimulate apoptosis of this cell. And this is very important to understand. So in our organism, every cell is under the control of our immunity. And to control every particular cell, immunity should not enter the cell and check what proteins are produced there. Instead, every cell is equipped with a special mechanism presenting examples of all its proteins on its surface. So by scanning the surface of the cell, our immunity can make a conclusion whether this cell is normal or is there any mutation. And as a result, killers just either leave this cell alive, or in case of any abnormalities, they might kill the cell. So type 1 of MHC class 1 receptors, they help to control our own cells. They help killers to decide whom to kill, whom to leave alive. Then type 2 receptors. Type 2 receptors are present on the surface of immunocompetent cells, and we call them antigen-presenting cells because they expose on their surface fragments of peptides which were phagocytosed by them. So the difference between the first and the second class of receptors is that type 1, they present their own peptides, examples of proteins produced inside the cell, while type 2, it presents fragments of peptides that were phagocytosed. These are foreign peptides. These are antigens which should be rejected by immunity. And type 2 receptors, they are present only on the surface of macrophages. And now we have to remember that the main essence of macrophages is not only in phagocytosis of the antigen, but just after phagocytosis of the antigen, macrophages expose those antigens on their surface to activate T cells and to launch the adaptive immunity. And that's what we are going to talk further. So now we have to say that MHC type 1 is recognized by CD8 killer cells. From the lecture blood, you remember that we have two types of T cells, CD8 killers and CD4 helpers. And now we are going to understand why we need those CD8 and CD4 receptors. So MHC type 1, here the cell, and here it's MHC type 1. It is destined, as we have said, to present its own antigens to killers. So killer decides whether to leave the cell alive if it produces normal proteins, or to kill the target cell in case of mutated proteins. So how it works? Peptides presented by the cell are recognized by a special T cell receptor, and at the same time MHC type 1 is recognized by the CD8 receptor, and that's what we call the double recognition event. So killers, they analyze peptides only in case they are presented by MHC type 1. And it is very important to understand. So double recognition, it means that CD8 killer cells, T killer cells, they scan the surface of every cell in our body by analyzing peptides presented by MHC type 1. Because if T cell receptor just recognizes antigen, but at the same time CD8 doesn't find MHC type 1, then this process is ineffective, it doesn't work. This interaction between the target cell and killer works only in case the peptide is presented by MHC type 1. So double recognition takes place. T cell receptor recognizes antigen, CD8 recognizes MHC type 1. And as it is written, if target cell produces defective proteins, the killer cell will stimulate its apoptosis. So after this interaction, killer cell decides whether to kill target cell or to leave it alive. Do not forget that T cell receptor is individual for every T cell, and that's what we are going to discuss later. Then MHC type 2, it presents foreign peptides to helper cells. As you can see in this case, almost the same interaction is shown. As you can see, peptides are recognized by the T cell receptor, here this foreign peptide and T cell receptor, while the MHC type 2 is recognized by the CD4 receptor. Again, double recognition takes place. So CD4 helper cells, they interact with antigens, with foreign antigens on the surface of macrophages or dendritic cells. And as a result, if they interact properly, then they get activated, increase in the number, and further stimulate proper B cells to produce antibodies against this antigen. So in this process, it is very important that T helper cell, CD4 T helper cell, could be activated only in case of double recognition, in case its T cell receptor recognizes proper antigen and its CD4 at the same time recognizes MHC type 2. So you have to know and explain what is the double recognition event. Then what is written, then cells bearing are called antigen presenting cells. Yeah, so only antigen presenting cells could activate CD4 helpers. Those antigen presenting cells are macrophages, dendritic cells, and B cells. Okay, let's move on. And here the same events are shown in a diagram. For example, the cell produces insulin, yeah, and examples of those insulin molecules are presented with the help of MHC type 1 on the surface of the cell. So killers, by passing by, they analyze the surface, and by analyzing the surface, they know everything about the protein synthesis inside the cell, whether it is normal or abnormal. And here we see MHC type 2 receptors. They present on the surface peptides engulfed previously by phagocytosis. As you can see, phagocytosis takes place, then in the endosomal compartment, those peptides are broken down into fragments and presented with MHC type 2 on the surface. That's the essence of macrophages. They not only phagocytose antigen, yeah, but they expose examples of those antigens on its surface, and in this way activate T cells. And I want to conclude, just to make one conclusion, I want to highlight one aspect. Notice that T cells cannot, let's move back, T cells, they can't be activated by antigen itself. So to activate T cells, antigen presenting cells are needed. Okay, this is very important. T cells, they can't decide what antigen to fight against. To fight against any antigen, this antigen should be presented by the macrophages, dendritic cells, or B cells. This is what we have to comprehend. Let's move on. Now, as for the T cell receptor, what is it and what is the function? T cell receptor is individual to every antigen. And now we have to say that around us in the outer environment there are many harmful antigens. Each has its own conformation. And to fight against them, we need to have a special T cell receptor with a complementary conformation. And this is the essence of our adaptive immunity. So T cells, there are lots of T cells in our organism, but every T cell is individual. Every T cell bears its individual receptor. And every T cell can fight against only the same antigen. So when organism is infected with exact antigen, only one type of T cells get activated and fight against it. Actually, not one type, because we are usually infected by bacteria which has many antigens and many respective T cells get activated. But that's what you have to understand, that not all T cells get activated. Only a set of T cells with their T cell receptor fit for this antigen. And how to say, this receptor should be relevant, should be fit for the antigen. Okay, then so all the T cells, they differ because every T cell exposes its individual T cell receptor. This T cell receptor interacts only with the proper antigens. That's what we have to understand, because further we investigate thymus, and we have to understand how those T cell receptors are formed and selected. Then, as for the B cells, the same way every B cell exposes receptors to only exact antigen. So despite we have lots of B cells, every type, when infection is launched, only a set of B cells work. Only those B cells with their receptors fit for the antigen, with the receptor which is cognate to this antigen. I want you to remember that B cell receptors is the same as antibodies, the same as immunoglobulins. So B cells, they produce antibodies in the outer environment to neutralize the antigen, and at the same time they use those antibodies as receptors. So here antibody is shown. This is the typical structure of the antibody, and examples of those antibodies are fixed in the membrane of the B cell. So B cell exposes its individual antibodies on the surface, and once again every B cell is individual. It has antibodies against exact type of the antigen. And as it is written here, every B lymphocyte has individual receptor specific to only one antigen. Interaction between B cell and proper antigen stimulates B cells to proliferate. So when we deal with exact infection, if infection is caused by antigen which is recognized by this particular antibody, such B cells get activated to produce more antibodies. So they start to proliferate and increase in the number. Then activated B cells are transformed into plasma cells and produce their receptors, antibodies, immunoglobulins to neutralize this antigen. And that's what we are going to discuss in the next lecture. And to complete our basics of immunity, we have also to say that there are different set of antibodies. First of all, during the first contact with antigen, immunoglobulin M are produced. Such type of immunoglobulins, they have low affinity to the antigen. They recognize the antigen, but not strongly. But at the same time, they are pentameric. So every immunoglobulin M is represented by 5 antibodies. So it has 10 sites to recognize the antigen. So they have low affinity, but they could be produced immediately just after the contact with the antigen, and they have lots of sites to interact with the antigen. After that, immunoglobulins G are produced in a week, after 7 to 10 days, and such type of immunoglobulins, they are monomeric, but they are highly specific to the antigen. They are much more effective in fighting against them. That's why usually after 1 week of infection, patient has the patient get better because of immunoglobulins G, because they are much more effective and they help to deal with bacteria. Okay, so immunoglobulin M, then G, then immunoglobulin E. These are immunoglobulins which are donated to basophils, to tissue mast cells, because mast cells, they don't know when to release their granules, their histamine and heparin. That's why B cells, they produce immunoglobulins E specific to the antigens they recognize, and they give such immunoglobulins to basophils, to mast cells. And mast cells, they use those immunoglobulins as receptors for antigens. And when antigens are recognized by those immunoglobulins, they realize their function, release histamine and heparin outside. Also, immunoglobulin D, they are used as receptors, and immunoglobulin A should be mentioned because it doesn't launch inflammation, but at the same time it is present in our mucosal envelopes, in epithelial tissues, in barrier zones, so they try to neutralize antigen before it enters our tissues, so it doesn't launch inflammation, but at the same time it is this is the dimeric molecule, it is represented by 2, and it works in our mucosal envelopes. Okay, this is what you have to know before we start to investigate lymphoid organs. And now let's start exactly from this slide. Lymphoid organs, this is the part of our immune system, and they are divided into central, what we are going to talk about this lecture, and peripheral. This is the topic of the next. Central lymphoid organs, they differ from peripheral, as here lymphocytes are produced and selected. So here lymphocytes, they do not perform their function, they are only produced, proliferate and get selected. In the peripheral organs, lymphocytes do interact with antigens and they perform their function. So here we say that antigen-dependent differentiation of lymphocytes takes place. So here lymphocytes, they interact with antigens and fight against it, while in the central organs, lymphocytes do not contact with foreign antigens and only maturation takes place. Okay, so let's talk about red bone marrow and thymus. First of all, red bone marrow, we have already considered it in the topic hematopoiesis, because this is the site of lymphopoiesis, myelopoiesis, and all leukocytes are formed there. That's why, of course, red bone marrow is the central organ of our immunity. If red bone marrow is damaged, then organism doesn't produce leukocytes, so immunity doesn't work at all. That's why it is considered as a central organ, because there leukocytes are formed. Then, what about the stroma of the organ? The three-dimensional scaffold is represented by the reticular tissue and mesenchymal stem cells. Besides, I think mesenchymal stem cells should be mentioned, because red bone marrow is typically associated with the hematopoietic stem cells, but also this is the rich source of mesenchymal stem cells. We have discussed them last time in the topic connective tissue, so if you are interested, then return back. Then parenchyma of the organ, or the functional elements, are represented by hematopoietic cells, including hematopoietic stem cells, of course. And what about the functions? Of course, here B cells, T cells are formed, as well as granulocytes. Then we consider now only those functions associated with the immunity. So as the central immune organ, the red bone marrow performs the function of antibody production, because after acute inflammation, after acute infection, for example, those B cells producing antibodies actively, they migrate to the red bone marrow, transform into memory B cells, and till the end of their life they produce these antibodies in a bloodstream. So we could understand whether this patient had already contact with this particular antigen by taking a blood analysis and detecting their proper antibodies. So after the first contact with the antigen, the plasma antibodies are produced till the end of the life. Exactly, they are produced in the bone marrow. And also in the red bone marrow, a reserve pool of neutrophils could be found. So those neutrophils could be mobilized in case of acute infection. Okay, we know that some neutrophils are associated with the blood vessels wall, some of them are fixed in the red bone marrow, and both could be mobilized to the bloodstream immediately in case of acute infection. So there are two storage of neutrophils. Okay, this is as for the red bone marrow and its function in immunity. And now let's talk about thymus. We have not yet considered it, and till the end of the lecture we are going to discuss it. Thymus, this is the central lymphoid organ, because here T lymphocytes are differentiated and get mature. Then, parenchyma of the organ consists of T lymphocytes, which are also sometimes called thymocytes. So if you see the term thymocyte, you should associate it with the T cell. And stroma of the organ is very unusual, as it is represented by epithelial reticular cells. Why they are called epithelial reticular cells? Because they derive from the epithelial tissue, while they perform the function of reticular cells. It is very important because you remember our epithelial tissue usually forms the covering, so it usually covers or lines our surfaces or cavities, or form glands. But it could form glands, but in this case epithelial tissue form three-dimensional scaffold, sponge-like structure, which is very unusual for epithelial cells. That's why I want to highlight the stroma of thymus is represented not by reticular cells, because reticular tissue is a part of connective tissue, quite another tissue. Yeah, but in thymus, stroma is represented by epithelial tissue. It's very unusual. That's why please remember. And that's why these cells are called epithelial reticular, because by derivation they are epithelial, by the function reticulum. Okay, then thymus is divided into lobules. Actually they are not true lobules because they are not fully separated one from another, but almost separated. Between those false lobules there is a loose connective tissue with blood vessels and nerves. Last lecture we discussed that this tissue is ubiquitous and it is present in every organ. And those lobules are divided into the cortex, more dark region, and lightly stained medulla, which is centrally located. And the difference is in the color staining is due to the concentration of lymphocytes. In the cortex, lymphocytes are much more numerous and much more tightly packed comparing with the medulla. Besides, I forgot to say that thymus is located behind the sternum above the heart, and this is the typical position. Okay, let's move on. And as for epithelial reticular cells, there are 6 types of those cells. If you want to get a good mark, you have to know. So type 1 of those cells, they line the outer border of the thymic lobules. So type 1 cells, they perform the border function and they contact one with another with the occluding or tight junctions, highlighting its insulatory role, its barrier role. So it insulates thymus from outer tissue, from the connective tissue capsule. After that, type 2 cells, these are the cells present in the thymic cortex, and they are unusual as they expose both MHC type 1 and type 2. So they could present their self molecules as well as molecules engulfed by phagocytosis. And those cells, they participate in the selection of T lymphocytes. So we are going to discuss their function next slide. Then type 3 and 4, these are the cells forming the barrier between the cortex and the medulla. So again, the exposed occluding junctions or tight junctions, and their main function is just to, to border two regions. Then type 5 of epithelial reticular cells, they work in the medulla and they participate in the further selection and differentiation of T lymphocytes. So this is the function. And type 6, these are the cells forming concentrical structures of epithelial cells, which are called Hassel's corpuscles. Hassel, this is the name of the author. So those corpuscles, their functions are not very well known. We know that their number increases with aging or with accidental involution, and they also produce some hormone-like structures affecting the differentiation of T lymphocytes. Okay, so now let's talk about the T cells education, or it is better to say T cell selection in the thymus, because this is the most important function of the thymus, to select proper T lymphocytes. Now we have to say that along with the maturation of lymphocytes, their T cell receptors are randomly formed. So by mistake, some T cell receptors, they could recognize our own antigens and start to fight against our own cells. So such T lymphocytes should be eliminated, should be killed. And this is exactly the function of thymus. Here, T lymphocyte, they start to differentiate. They start to expose their receptors, which is randomly formed. And in case this receptor starts to fight against antigens of our own organism, thymus eliminates them. So in this way, 98% of all T cells are destroyed in thymus. So the selection is very strict, and let's discuss it stage by stage. So the first stage of T cell differentiation arrives from the red bone marrow. If you remember the topic hematopoiesis, you remember that our hematopoietic stem cell splits into two: myelopoietic precursor and the precursor for lymphopoiesis. And exactly this second stage, this precursor for lymphopoiesis, arrives to the thymus, and we call this stage double negative stage. Why double negative? Not because it is negatively charged, so there is no any charge on its surface, but because it has no CD4, neither CD4 nor CD8 receptor. So double negative because it is neither helper nor killer at the same time. It is devoid of both receptors, so it is immature T cell. And those cells, they occupy the subcapsular zone here under the capsule. They are located and they start to proliferate, just they simply increase in the number. After that, the second stage is called double positive, because every cell descending to the cortex start to expose both receptors, helper CD4 and killer CD8. So at this stage of their development, they are killers and helpers at the same time. And in the cortex, the positive selection takes place. And this is the key event in the selection of T cells, and that's what you have to comprehend. You remember that here, epithelial reticular cells type 2 are present. They contact one with another with the help of desmosomes. They create a three-dimensional scaffold, and they interact with those lymphocytes, and they expose both MHC type 1 and MHC type 2, both receptors on their surface. And here, CD4 receptors, they interact with MHC type 2. CD8 receptors, they interact with MHC type 1. And as a result, if this interaction is proper, then such lymphocytes, they survive. So here we check their possibility to perform their key function, to interact with MHC receptors. And for this purpose, epithelial reticular cells, they expose both receptors on their surface to test both CD8 and CD4 receptors on lymphocytes. So here the positive selection takes place. Why it is called positive selection? Because in this process, if CD4 properly interacts with MHC type 2, or CD8 properly interacts with MHC type 1, in this case cell survives and descends to the medulla. In case this interaction is improper, in case CD4 or CD8 is defective, or T cell receptor prevents proper contact with the MHC receptors, in this case such lymphocytes are destroyed with the help of apoptosis. because such lymphocytes, they can't perform their function. We don't need them. If they can't interact with MHC, this is their critical function, then we don't need them. And this is exactly what is tested in the cortex, okay? So here, epithelial reticular cells type 2, exposing MHC type 1 and type 2, test those CD4 and CD8 receptors. And if interaction is proper, such cell is confirmed its quality, yeah, and it descends to the medulla. It continues to develop. In case the interaction is improper, it is destroyed. That's why cortex is more darkly stained than the medulla, because here we have many more T cells than in the medulla, because they are killed predominantly in the cortex, and only several of them descend to the medulla. So next, what happens in the medulla? Next step of T cell selection takes place. Here in the medulla, we have single positive stage. So cells, they lose either CD4 or CD8 receptor. So they now are divided into two groups, killers and helpers. So in the medulla, they are almost mature and they expose on the one receptor. They are specialized. They've already chosen their fate, and they decided whom to be, helper or killer. And next stage, it calls the negative selection. Negative selection. As it is written here, type 5 epithelial reticular cells work, and type 5 cells, together with the dendritic cells besides, they also expose MHC type 1 and 2 on their surface. And here they check how strongly T cells do interact with MHC. And if this interaction is too strong, if after interaction T cells, they can detach, if this attachment is irreversible, for example, such lymphocytes are destroyed. So if in the cortex they had to interact with the proper receptors just to survive, here they need not only to contact, but also to demonstrate that this contact is not very strong, that it is reversible. Because in future, when they perform their function, they need to check the quality of the receptor and after that detach and check the quality of many other receptors. If they contact with the receptor too strongly, then they could fight against our own cells. So such cells are destroyed, and that's what we call the negative selection. So here, if interaction between MHC and CD receptor is too strong and firm, such T cell receptors connect to strong. Such cells are killed by apoptosis. That's what we call negative selection. Why it is negative? Because if cells recognizes something, then they are destroyed. Positive selection is when they do something, they survive. Here, if they do something, they are killed. That's why the process is negative. So these are the two slides you have to comprehend to understand the function of the thymus. So thymus helps us to eliminate all T cells with their T cell receptors destined against our own antigens or produced against our own antigens. In this way, thymus helps us to avoid autoimmunity. And that's why without thymus, our adaptive immunity is impossible. Impossible because T cells are generated predominantly against our own antigens, and without thymus they can be eliminated. So without thymus, our immunity would work against our organism, and only thymus is needed to prevent that, to eliminate all T cells destined against our receptors. Okay, here is the very helpful video, and the hyperlink will be in the description below. Then also we have to mention that in the thymus we have a blood-thymus barrier. Blood-thymus barrier means that blood vessels in the thymus, they are equipped with additional envelope, preventing the contact between the blood-borne antigens and thymic T cells, immature T cells. Before becoming mature, before they complete the process of their maturation, T-lymphocytes should not contact with blood, should not enter the bloodstream. So inside thymus, a proper microenvironment should be maintained. And to insulate lymphocytes, immature lymphocytes from the bloodstream, there are several layers. First, we have to mention that there is the endothelium. After that, basement membrane with the pericytes around them. After that, perivascular connective tissue. Here it is with macrophages inside them. And after that, that's what's unusual and what is present only in the thymus. This is the basal lamina. Here, this is the red one. Basal lamina and epithelial reticular cells. So those two components are absent in other organs. In other organs, blood capillaries are lined only with the endothelium and basement membrane. In case of thymus, every vessel additionally has epithelial reticular covering. Why? Because if foreign antigens coming from the blood affect lymphocytes, they might, they might disturb the process of their maturation. And opposite is true. If immature lymphocytes would enter the bloodstream, they might fight against our organs. So they might launch autoimmune conflict. That's why lymphocytes are held inside the thymus and prevented from entering the bloodstream before they get fully matured and before they undergo all the process of selection. Another term we should mention in thymus, this is the age involution. So thymus is an unusual organ also because it starts its back development early, just after the puberty. So as it is written here, maximum size of the thymus is achieved at puberty, and then the process of age involution is realized with the replacement of cortical thymocytes with fat and increased number of Hustle's corpuscles. It is easy to see looking at this diagram. It is easy to understand. As you can see, at the moment of birth, here we have this ratio between the medulla, cortex, and fat. So cortex and medulla, they prevail. But starting from 20 years, we can see decline in the amount of cortex and medulla and the replacement with the fat. And at the age of 35, almost all thymus get replaced with the fat, this green tissue. And here how mature thymus looks like at the age of 30. Compare with a newborn child. So there is nothing maybe unusual because all organs with aging they get replaced with a fat or connective tissue. But what's unusual with thymus that the involution starts very early, just after the puberty. Why thymus undergo age involution so early? That's because we investigate our outer environment very actively just after the birth. We contact with thousands, millions of new antigens every day of our life. But at the age of puberty, it is considered that we are already familiar with almost all antigens. Something like our vocabulary. Just after the birth, we investigate new words every day. But when at the age of puberty, we are already know almost all the words we need to explain what we want. The same is true for antigens, and it is considered that after this age we do not contact with lots of new antigens, that almost all antigens are already familiar. That's why we don't need thymus. And if thymus doesn't undergo age involution, this is the problem, and this is the sign of hyperactive immunity, which is the same problem is not effective or low activity of immune system. Okay, along with the age involution, we have to know what is the accidental involution. This is another process which is realized by accident in case of stress, for example, severe diseases, infection, or different other interventions. And due to the stress, we can see high concentration of corticosteroids. These are hormones produced by adrenal gland. And as a result, due to stress, T-lymphocytes, they are massively destroyed. And as a result, there is no difference between the cortex and medulla in our thymus. As you can see, almost the same cortex and medulla, the same color. That's how we can distinguish accidental involution from age involution. In age involution, in case of age involution, thymic globules will be replaced with the adipose tissue, while in case of accidental involution, there will be no border between the cortex and medulla due to massive destruction of T cells inside the thymus in response to stress conditions. Okay, and at the end of our lecture, we have to say that thymus derives from our pharyngeal region, and also different other organs do derive from this region. And we have to say that here we have pharyngeal pouches. Pharyngeal pouches, these are invaginations of our endoderm directed towards the ectoderm. And at the same time, we have pharyngeal clefts, which are formed by the ectoderm. So please do not confuse, and it is very important, and it is asked in many MCQs to confuse you. And many students get confused because pharyngeal pouches, these are invaginations of the endoderm, while pharyngeal clefts, these are invaginations of the ectoderm. They are directed one to another, and in fishes, gills are formed in this place, so they get fused, forming gills. In humans, they do not fuse, but those invaginations, they give rise to many different organs. For example, the third pharyngeal pouch gives rise to the thymus and parathyroid glands. Notice that thymus is formed from our endoderm, from the third pharyngeal pouch, and it migrates down and stops behind the sternum. Why it is important to know? First of all, because sometimes migration is violated, so thymus might stop in the neck, not reaching the sternum or thoracic cavity. Sometimes thymus is migrated together with a parathyroid gland, because parathyroid gland is also formed from the same source, from the third and fourth pharyngeal pouches. So sometimes thymus get fused with a parathyroid gland, involving it to the thoracic cavity together with it. And what else? Again, notice that the stroma of thymus is represented by the endodermal epithelium, which is very unusual for epithelium to form the stroma of the organ. Then, what we can see in the thymus is the specimen, what we ask you during the practical lesson. So we can see the capsule, connective tissue capsule, here it could be found. Then those lobules, which are false lobules, as you can see, they are not fully separated. Yeah, the connective tissue invades the organ but doesn't separate it fully. And then inside those false lobules we can distinguish cortex, which is much more dark than the medulla, which is much more lightly stained. And now you know why, because almost all T cells are killed along with the process of positive selection in the cortex, and only a few of them survive and enter the medulla. Okay, in the medulla also we can see large blood vessels and some Husserl's corpuscles, but I can show you with this magnification. Okay, and at the end of the lecture I want to ask you to answer the question from the USMLE database. It is difficult, but let's try. First of all, here we have 4-month-old girl, and she's frequently suffering from the respiratory infections. So this is the 5th time her daughter has had an upper respiratory tract infection. So this is the sign of immunodeficiency. At the same time, notice that her medical history is also significant for seizures shortly after birth. Also, she has elevated temperature, so again, it seems that she has an infection. And both ears are slightly malformed. And the question is, what organ is malformed, and what is the source of this organ development? And the answer, I think, is very easy for you, so you can guess easily, because we have discussed only one organ and you know the source of its development. But I want you also to give an answer. What syndrome is this? How we call this syndrome when this organ is malformed? What genetic pathology it has, this girl has, and why she has these seizures, why this syndrome, this immunodeficiency, is accompanied by seizures shortly after the birth. So please, let's try to reveal together your answers. Please put in the comments below, and I'll be very glad to read them. Okay, good luck, so we'll see you next time.